Okay, so we will go ahead and get started. This is awesome. This is definitely, I think, the most attendance we've had at an event since my time here, a couple of years. On top of that, definitely the most attendance at our first event of the year. So thank you all for being here, whether that was bribery or not. Hopefully you're going to learn something. Hopefully you'll enjoy it. And understand that these are all opportunities that these students went and found either on their own or by asking a professor. And they just basically said, this is the career I want. This is the field I want to work in. These are the opportunities I need to do now if I want to determine A, do I actually want to do that? And B, am I actually interested in it as well? And am I in the right spot? So without taking up any more of their time, I want to turn it over to our first student. This is Kaitlin Koster. She is a junior biology major, and today she is going to be telling us about the population ecology of Mooneye, species association, and assemblages.
Hi guys, as you said, I am Kaitlin Koster. I'm a biology major here, and this is what I did last summer. I got to spend my summer in Potsdam, New York at Clarkson University as a part of their Asset RAU program. The project I worked on was called Population Ecology of Mooneye, Hiodon tergisus, Species Association and Assemblages under Dr. Tom Walland. So let me introduce you to the Mooneye. They are also called Hiodon tergisus, which roughly translates to toothy tongue, which as you can see in this bottom picture, they have cartilage on their tongue that kind of forms these little tooth-like shapes. They're kind of wild. So that tells them apart from other fish, as well as the big moon-shaped eyes, hence their common name. Their status is threatened in New York as determined by the New York State Department of Environmental Conservation. And there's really not a lot of research on these guys. So our overall goal for the summer was to create a species distribution model for predicting their populations. A species distribution model takes different environmental factors and different population data, kind of layers them on top of each other in a GIS-based system in order to take known populations of a critter and predict other ones based on it. So their distribution, as currently known, is from the Hudson Bay Basin to the Great Lakes Basin, the St. Lawrence River, and the Lake Champlain drainage basin. As you can see on my map up here, that red circle is the St. Lawrence River and our main focus for the summer, specifically the Oswegatchie and Indian rivers. Now, if you've never seen the border between New York and Canada, it's kind of a rough map, but up here is Canada, and this is New York. There's a line that goes through that represents the St. Lawrence River, which is kind of the border, the divide. All of these rivers drain from the Adirondacks into the St. Lawrence River, and in my blown up map, you can see the blue line is the Oswegatchie River and our 3 sampling sites on that river. And then the other one down here is on the Indian River. So my hypothesis for this summer based on basic life history research was that I expected the Mooneye to be associated with fish that are found in fast-flowing, clear and open waters of medium to large rivers with gravelly riverbed. If this hypothesis is correct, presence of these associated species can be used to locate other areas possibly inhabited by Mooneye. And then I also made some predictions. So those fish bolded with green are pictured below if you're interested. But I expected some fish like lake sturgeon, quillback, redboards, northern pike, muskellunge, perch, smallmouth bass, walleye, and fallfish based on also similar life history data. So our methods this summer were based around our electrofishing research vessel, pictured at the top, lovingly called the E-boat. This E-boat has 2 arm-like structures called booms that dangle our anodes into the water. These anodes send out an electrical impulse about 6 to 8 feet in every direction that kind of circles around and comes back to the hull of the boat where the cathode is. When the fish feel this electric shock, they're kind of stunned for a minute, so they kind of freeze, turn belly up, and float to the surface, where my research partner and I are standing on the front of the boat with really long dip nets, so we can either identify the fish or scoop up the ones that we want. The fish are not permanently harmed. They recover really quickly. So when we did catch a Mooneye, we would obtain data like length, sex, and weight, and also some scale and fin samples. The scale and fin samples were interesting because fin samples, as you probably expect, used for DNA data, but the scales are really cool because like tree rings, every year that a fish is alive, they gain a new ring in their scales. So we were able to age them using this. And we also would implant a pit tag, a passive integrated transponder that would allow us to see if we ever recapture these fish. We also kept specific count on what fish we saw while electroshocking so that I could perform my assemblage analysis, which I'll talk about in our results. We caught 8 total Mooneye this summer. So it turns out that threatened analysis was actually pretty accurate. So can't do much with 8 data points. So I reached into some old data from 2021 and 2009 and ended up using about 26 total Mooneye for my analysis. So to start my analysis, I ran a chi-square contingency analysis on the assemblage of fish when Mooneye were present and Mooneye were absent. I found no significance, which I'll talk about a little bit more later, but as you can see, for a chi-square of 18.5 on 14 degrees of freedom, a p-value of 0.19 is not what we're looking for. In stats, we're looking for a p-value of 0.05 or lower, so 0.19 was not doing it. So I didn't lose hope. I switched to an individual sort of analysis. This is called a Fisher's exact test. In this specific instance, this was going to be a little bit more powerful for me when I was analyzing the specific fish versus the Mooneye. So I used a 2x2 contingency table to create this data. And here were my results. As you can see in this column, these are all my p-values, and I had one significant p-value. My brown bullhead was significant with a p-value of 0.018, which we'll talk about a little bit more in a minute. This graph kind of shows you a little bit better the difference we were looking for. So this dark green color is the percentage of trips where we caught both Mooneye and the fish pictured on the bottom, written on the bottom. And the lighter green is when Mooneye were not present, but we did catch the fish. So what we're really looking for here is a difference, a big difference between the bars. And you can see pretty clearly this brown bullhead has a very large difference. So what does this mean? Overall, we had too few data. We were able to draw one conclusion, that the brown bullhead are negatively correlated with the Mooneye, which means that if Mooneye are present, bullhead are not going to be, and if bullhead are present, Mooneye are not. This isn't surprising. They have very different life history data and different riverbed preferences. The bullhead tend to prefer more vegetated area, more stagnant, warm water. And as I mentioned, the Mooneye prefer like that clear, that cold, but fast moving water. So my assemblage that I created, although it wasn't a significant assemblage in relation to times we didn't catch Mooneye, it was still a valid assemblage that agreed with previous studies done in the area. The issue with this assemblage is that all of the locations that we sampled this summer were chosen because Mooneye had been sighted there before. So it was kind of difficult for me to go in and compare an assemblage with Mooneye, where Mooneye had been sighted versus not sighted, because they'd been sighted. As I mentioned, too few data, so we were not able to yet create the species distribution model. However, this will be created in some coming years. And this is a picture of the bullhead. So my conclusions and next steps. This summer, in my analysis, I only used data collected by boat electrofishing. There's a lot of different ways to catch Mooneye, such as gill netting, angling, backpack electrofishing, but in order to reduce my confounding variables, I just stuck to data collected by boat electrofishing. And in previous years, it has been determined that this is the most efficient and most humane way to catch them. They used gill netting a lot last year, and it had a lot higher of a death rate for our Mooneye, and that's not the goal. As I also mentioned, we need more data for the species distribution model. However, this study did give us a valuable layer to the model, because if there's a prominent brown bullhead population in one area of the stream, there's not likely going to be many Mooneye there. So it did give us some valuable feedback. For future research, obviously more locations, somewhere Mooneye have not been sighted, so that this assemblage can actually be put to the test, and we can actually see if it's accurate for the species surrounding Mooneye. We also need to do more environmental factor monitoring. As I mentioned, species distribution models require 2 parts. We spent a lot of time on our population data this summer and not a lot of time on our environmental factors. They require both. So we need some more environmental factor monitoring and some more bed data. Actually, right as I was about to leave, we got some cameras in the mail that can be mounted to the front of the boat and can monitor that underwater data while we're electrofishing. Overall, conservation requires management. More data is always needed, and there are so many species out there that are threatened and endangered that need someone like us to go out and do research on them and figure out how to manage them. Also, the most fun part of my time this summer was all the fishes, so I thought you might want to see some more pictures of fish. This is a muskellunge. This is the biggest predator fish in the St. Lawrence River. He's the only one him we caught this summer, and he was our biggest fish. That is an American eel, a freshwater drum, a chain pickerel, a sea lamprey, which are both invasive and parasitic. We were not expecting to see him. Rock bass, they have these really vibrant red eyes that are kind of unnerving to look at. A carp, also invasive. A walleye, we caught the most of him. And look at those chompers, gives me every time. Bluegill, very common. Pumpkin seed, probably the most vibrant we caught this summer, also very common. And my favorite, the long-nosed gar. Look at those polka dots. He doesn't even look real. Okay, acknowledgements. Of course, I have to thank my fantastic research mentor, pictured at the top middle, Dr. Tom Langan. He provided a lot of guidance and a lot of knowledge this summer. We learned so much. Angelo Struche, pictured on the right in the top picture, he was my research partner. He goes to the University of Puerto Rico, and he did his on ecomorphology of the Mooneye. So if you're interested in that, come see me afterwards. I'll direct you to this page. Dave Sumner, our graduate student. Ethan Bailey, our undergraduate student. Dr. Baki and Mr. Philip White created the co-PIs of the NSF Asset RU program, and of course, NSF themselves, because this would not have been possible without them. There's my lovely poster. Got to present her at a poster presentation at the end of the year. And last but not least, I kind of wanted to talk about my personal experience because I didn't just spend my whole summer on a boat. Like, I lived there for 10 weeks. So if you know me, you know that I was so scared. This was one of the biggest things I've ever done in my life. So I was terrified. But this experience helped me grow as a person, as a student, and a biologist. And I gained so many new experiences. Turns out you learn a lot when you step out of your comfort zone. So I made so many new friends, climbed so many mountains, ate so many trail PB&J, and went to so many cool new places. The top picture, I'll start there. That's a bog. If you ever get a chance to go experience a bog, it's the coolest ecosystem I've ever been in. We spent a weekend in Montreal. We spent a day fully going between all the dams and locks of the St. Lawrence River and learning about how they manage that. That mussel at the bottom, there's nothing special about it, but it represents aquasasne because a big part of this RU experience was spending time in aquasasne and with the indigenous Mohawk and Haudenosaunee people of the area and learning about their culture and their history. So that experience was super important to me. And then that far picture is a castle on one of the islands in the Thousand Islands region of the St. Lawrence River. So lots of cool experiences, lots of new things. Now, any questions about my fish, about my experiences, or about applying to RUs?
So hold on just a second with all of your questions. We're going to do questions at the end for all of the speakers. That way everybody has the same amount of time to actually present. So if you have a question for Kaitlin, go ahead and write that down, jot it in your phone. We'll get back to questions as a group at the end and you can speak to all of them. Thank you. Thank you, Kaitlin.
Okay, so our next speaker, I think she's been a part of a different project every semester I've been here, maybe even multiple. So she's constantly staying busy. She's always doing work. This is Carmela Lewis. She is a senior biology major, and today she is going to be telling us about her summer experience doing wildflower surveys in Walland, Tennessee.
So, hey guys, as said, I'm Carmela Lewis, I use she, they pronouns, and today I'm going to talk to you about my senior study that I got to do over this whole last summer. I started in the spring and actually just concluded my final data yesterday. So I'm going to talk to you guys a bit about what I got to do, all the fun I got to have, and yeah, if I speak, sometimes I get really excited. So if I speak really fast, don't be afraid to tell me to slow down. Okay. So I actually got to have my senior study that was a partnership with Mountain Homes. Has anyone here been to Mountain Homes? Yeah, there's normally a fairly good amount. Mountain Homes has worked really closely with Maryville College over the years. They're about 20 minutes away in Walland, Tennessee. And we have worked closely with them with stuff such as the American Chestnut Orchard that we have over there, as well as other senior studies. We had a senior last year, her name was Erin, who did some senior study with the American Chestnuts. There's people who live there full-time, not too many, and then there's also a lot of people who work there part-time. So they are actually also directly beside the National Park. So a lot of the research I got to do could suggest that the wildflowers I saw could be wildflowers that are also seen in the park and larger area. Oh my gosh. So here's a fun little part. If you saw it, no, you didn't. Does anyone happen to know what the flower seen right here is by chance? Is it an iris? It's an iris. This is actually a dwarf crested iris. It's the scientific name right there. This was one of the very first flowers I got to see. And a fun fact about this guy is that it has been used historically by indigenous peoples as a salve to treat ulcers. So all the wildflowers you're going to see on this slideshow today have been taken by me, observed by me, stuff like that. So my role, I was a wildflower surveyor. I didn't go in expecting to look at one specific species. Instead, I wanted to take a naturalist approach and gather observations about the entire environment that I saw around me. So my goal was to find wildflowers, identify what the flower actually was, determine if it was native, and then record some generalized information. That's all the stuff that's going in my thesis. But as part of my partnership with Mountain Homes, they also requested a small booklet. So in addition to my thesis, I'm going to be creating a booklet of all the wildflowers in their area, and I'll include stuff like the really cool uses that they have, symbolism, all different stuff that's been requested by the residents for their satisfaction. Does anyone happen to know what this guy is? Oh my gosh, Miss Kaitlin. Lady slipper. It is. This one isn't necessarily as fun of a fact, but an important fact to note about pink lady slippers is that they are actually very heavily hunted. They're considered to be a very beautiful flower, and so people will do a lot to get these in their gardens. I had to make agreements with some of the people at their cabins when I did record a location of a lady slipper, announcing that I wouldn't give the exact specific location of that lady slipper and instead a generalized area of it because it is so heavily sought. Right? Another really big thing I had to consider is what even is a wildflower? I knew looking into this and wanting to do this as part of my senior study that wildflowers in the area haven't been observed and really surveyed in over a decade. So I knew it was important, but I didn't really understand what a wildflower was. So in the most general, common term, a wildflower is just a flower that's not in active cultivation. So if you go along the neighborhood or if you walk outside the college and you see flower beds, beautiful flower beds in front of someone's lawn, that's a cultivated flower. You wouldn't consider that a wildflower. The wildflower packets you can grow in your yard technically aren't wildflowers. However, if you went out in the Maryville College woods and you saw a mayapple or a trillium, something like that, you could consider that to be a wildflower. Some people go a bit more specific, saying, hey, we don't include shrubs or trees or vines. Those aren't wildflowers. But from my perspective, I included the most general terminology just so I could give as much data to the Mountain Homes residents as possible to increase that customer satisfaction that I was having with them. So yeah, what was I doing actually? Well, twice a week, each week, from the end of February until yesterday, I was actually going to Mountain. One part of the week, I'd spend about 4 hours going along the main trail they have there called the Cane Creek Trail. This is about 3 miles. The other part of the week, when I'd go, I would actually examine 10 different cabins that were all strewn out across the property, and then a pasture as well. The pasture was being constantly mowed, so while we didn't get a lot of wildflowers we saw there, we did get to see a really cool bit of that beginning primary succession of watching which flowers or which grass overtook the area overall. So, yeah, does anyone happen to know what this cool little flower is? Okay, that's totally okay because I didn't either. This guy is actually a Virginia pennywort. Fun fact about them is they actually hold a really close symbiotic relationship with the fungi around them. This was one of the very first flowers I actually got to see that I didn't know what it was. So this was the kickstart to my process of actually figuring out how to identify it, where to go, stuff like that. So yeah, what was the process? I actually had to get to the location. I often, instead of taking the school vehicles, which I was allowed to, I take my own vehicle because one of my biggest helpers on this trip was my dog, Olive. So I take my dog Olive with me to go survey the location and collect all my data points. I made sure I had everything I needed, pretty much only being a GPS, my Write in the Rain notebook, and a really good camera. Then I'd walk the location. Afterwards, I'd observe the flower. I'd go and I'd take pictures, many pictures. If I try to open up the Google Drive I have of the pictures on my phone gets really hot and angry. So we don't do that anymore. Then I record the location with the GPS and I write those GPS coordinates in my notebook just to have multiple locations and multiple places that I have this data in case I were to lose anything. And additionally, I write extra information in the field notebook. If I didn't know what the species was, I wanted to write what type of leaves did it have? Did it have leaves only at the bottom, being the basal leaf? Did it have beautiful white petals? Did it have 5 petals? Did it have 7 petals? Stuff along those lines. Afterwards, I finished my whole survey for the day and I come home. The flowers I didn't know, I would upload those pictures to software and it would identify the flower to its closest, highest percentage that it could. Afterwards, I'd take those pictures and I'd open up my older books and like physically match up the picture with the software identification with what I had and make sure everything lined up so I could confidently say, hey, this is the flower that I did see. Then I would determine if it was native. This was a specific request by the Mountain Homes residents. They wanted to know the native flowers in their area rather than all of the flowers in the area. So my study only specifically includes the native flowers in the area, although I do have the non-native for anyone who is interested. And then finally, like I was talking about a bit earlier, I discovered more information about them. I'd include a little physical description of how to better identify them, as well as different uses that I've been sort of telling you guys about earlier. Is there a medicinal use, a historical use? Are they toxic to humans? And then a personal aside for me, I wanted to include symbolism. I think that's a really cool thing that's been used throughout history and talked about that has kind of been lost a little bit to time. I'd also include bloom times and then create a map of every single species observed with their locations that they were observed. Does anyone happen to know what flower this is? Oh yeah, a bloodroot. It actually is a bloodroot. And the fun fact about it is it gets its name from the sap that it secretes. If you happen to find a bloodroot in the wild, in order to break apart its stem or the roots, it seeps this very sappy, reddish orangey substance. That's also been used by indigenous peoples as dyes. Overall, I don't have an exact number because again, I finished just yesterday, but I've discovered over 100 species. That includes over 1,000 data points that I've collected in my GPS. So if I tried to show you my GPS, it would just be a very big blur of little flag marks that you couldn't really understand. I can't really believe it. So if you can either, totally understandable, but I got to see a lot of really cool things. I got to see wild geraniums. I got to see spotted trout lilies. I got to see fire pinks. Fire pinks are one of my favorite flowers because that was one of the first ones I got to see that was so bright red. Then I got to see things like flea veins, different types of spider warts. And around this time, I had started in February and I was maybe in March, end of April, had a chance to breathe. I wasn't seeing a lot of new things. And then I blinked. And then I saw tons of more things and I wrote them down and I ran out of room in my notebook and I got a new notebook. I had to get a new SD card for my camera. And thought things were calming down. And then I blinked. And then I saw even more, guys. I saw violets and cinquefoils and common elephant's woods, more fire pinks, beautiful cardinal flowers. And I kept going and seeing azaleas and heartly foam flowers, all different types of guys. And it really just kind of blew my mind. I've really been excited about wildflowers. That's one of the reasons I really wanted to do this, as well as get that naturalist perspective. But I didn't truly understand how little of the environment in the world I was seeing until I actually got to do this study and see over 100 different species of native wildflowers alone, not even including them. So yeah, that's about what I did this whole summer. Thank you guys for listening. I would especially like to thank all the people who went with me on my journey, including my beautiful dog Olive right there. I think I had my whole family at some point, my partner, my suite mates, everyone. It was a really great time and I'm very thankful for that. So yeah, I'll be happy to talk to you with questions afterwards.
All right, so we have our final speaker of the day. I want to introduce you all to Jessica Mohr. Some of you may know her. You may have known her as a computer science major. She is now a sophomore biochemistry major. And today she's going to be talking about her REU over the summer, the effects of adversarial attacks on deep learning models.
All right, so as Brett said, my name is Jessica Mohr, and I'm a sophomore here. And I'm going to talk to you about my time at Boreanell. If you're not familiar with Boreanell, it's the Oak Ridge National Laboratory. It's about 40 minutes from here. But here to start a little bit about me. I am a sophomore. I'm from Oak Ridge, so I got to live home over the summer. I've been very familiar with the lab, so it's really great to get to work there. I am in Guild and a science scholar here, so I know some good science scholars. And I am now in biochem with a 3-minute concentration. Normally you can see me and also my cat, because I'll take any opportunity to show a picture of my cat. His name is Dave. So a little bit about Boreanell. It is located in Oak Ridge, Tennessee, obviously. It was first built during World War II. If anyone saw Oppenheimer, they were the facility tasked with processing the uranium. Now it's kind of shifted over, and the site that I was on no longer is a processing site, but is dedicated to research. So they have over 5,000 scientists from 60 different nations, and there's work done in almost all STEM areas. So the one most commonly known would be like the chemistry, the atomic side, but they also have the computer science division that I worked in. So I was part of their AI and learning systems group. So my summer project was titled The Effects of Adversarial Attacks on Deep Learning Models, and I'll understand what that means. It's one of the big fancy words that I didn't really understand until I worked there for a little bit. So first I have to address what is machine learning. It's basically AI, which I think everyone's kind of heard about, stuff like ChatGPT, but the most common uses would be like self-driving cars, search engines, social media. The designer I used to make the slide in PowerPoint would also be a machine learning model. And we can basically boil down to models and algorithms that are used to power modern AI technology. You kind of see the parts of it. So an adversarial attack is an algorithm that applies a perturbation to an image, because my project used image recognition models. You're trying to feed an image to a computer and get it to say, like, this is a car, this is a cat. So you can kind of see there, you have the car on the left. That's just like a normal image that you give to the model, classifies it as a car. Then the noise in the middle there is the perturbation that's applied over the image. And so the one on the right is the modified image. The main issue here being that that car looks basically no different to the human eye, but it was classified wrong. I believe in this one, it said it was like a minivan. And so while it's still close, it's obviously wrong. And while in this case, classifying something as a sports car to minivan isn't necessarily that bad, if you're in your car and it says the stop sign isn't a stop sign, that's not gonna. So to counter these attacks, we have adversarial defenses. And the whole point of defenses is to mitigate the effects of the attacks we discussed. So you can use defenses on the inputs or the outputs to try to get a correct prediction instead of a wrong one. So the basic kind of diagram there is we have our bear and it applies the attack and now it thinks it's a horse. And we apply the defense and it goes back to bear. So that's kind of our goal with the defenses. So in my project, we use 3 defenses at the same time for all of our data, which just means that like instead of it being 1 defense for every test we ran. So kind of the methods we used was our goal was to measure the effects that attacks and defenses have on different like pre-trained models. There's some that are like easily available to people that already trained that anyone can kind of pull from the internet. So that would be really trying to train a car and all that taking a long time. So this is the graph of the baseline. You can kind of see the bottom, the x-axis is the correct label, and the y-axis is what the model predicted after you got an image. So the graph line at the bottom shows that they were correct most of the time when there were no attacks or defenses. I think this was something like 80–90% accuracy of the pre-trained models. And then the results after the attack are obviously significantly more spread out. It's very colorful, fun graph. I've had fun with those. So an important thing to note about these is they're untargeted, and that's just a distinction. That means that the goal of the attack was to just change her to a wrong image. It didn't care what that image was. So we could go from cat to dog, from cat to horse, and it would still count that as a successful attack. So these are 2 different attacks here. They're called projected gradient descent and square attack. And so you see the one on the left is the projected gradient descent. That one was significantly more effective, at least on the models we used. And then the right one is square attack, which was not as effective. You can see by the lack of distribution there. And the important thing to note here is that if you look at these graphs, kind of hard to point to on the PowerPoint, but there are certain lines that kind of emerge of classes that the model preferred to predict after they were attacked. So for at least for my research, it commonly predicted birds, mammals, and bugs, which we believe was part of the data set because the data set we used started off with a bunch of animals and things like that. So what we kind of saw was that the average accuracy following the attack and defense pair dropped to about 50% instead of 80%. So that was not as significant of a drop as I think we were initially expecting, but still not great. Only about 50% of the time. And the attacks did cause models to favor certain classes. So in a larger study of this stuff, probably you would see different favored classes, but we believed that it probably would favor certain ones every time. And the classes vary slightly. So what we can do with this is look at the classes that are preferred in a bigger study and see like what's similar between them so we can better develop the defenses against these attacks. And the one personal takeaway I wanted to include, because as Brett mentioned, I'm now in biochem, was for any freshmen or sophomores here, try to get practical job experience before you graduate. Because I love my computer science coursework and I love computer science clubs, but I went and I did the job and I decided that's not what I want to do for the rest of my life. So I would recommend that just so you don't get out of here with a degree and then decide that's not what you want to do, because any experience is going to be very different than a real new class. So yeah, thank you guys for listening, and if you have any questions, you can email me or call email there.
We'll go ahead and bring the other speakers back up. Before we get to questions, I want to go ahead and let everyone know, we do have 7 more of these events this year. The next event that we have coming up is going to take place on September 19th. So that is going to feature 2 alumni, Stephanie Collins and Whitney Hall Vesser. Stephanie is an analyst at KUB, and Ms. Vesser is a water quality lab supervisor.
That sea lamprey that you said was invasive. I'm so sorry, please again. The sea lamprey. What exactly is it, and did you get rid of it?
So it's a parasite, so it has, its mouth is like circular, and it kind of, if you've seen Stranger Things, it kind of looks like a demogorgon. It's wild looking. And yes, we were supposed to get rid of it, but it was like our first trip, and we didn't think about getting rid of it. We were supposed to, but we did not.
So you're just like,
yeah.
you said that you used some sort of software to identify the wildflower. What did you use, and how accurate was it?
Oh my gosh, thank you for asking. So I actually used a lot of apps that were on my phone. A lot of the areas I went to didn't have service, but I could take pictures and then look at it later. I used 2 main ones, being iNaturalist Think, which is a really good app, it's free to use, and another one being called Picture This. Now, there actually were some studies done that showed that Picture This has a 98% accuracy rate compared to Seek and another one known as Leap Snap. So Picture This was the primary one that I primarily initially uploaded everything to, and then afterwards I went and checked with the other 2, and then finally my books.
Can you talk a little bit about your role in what, were you looking at the data that was coming out of these things, or were you developing the defenses, or? So my role was, I initially worked to write the programs that would take the data, and then I was looking at them. I myself did develop any facts or defenses because that gets very technical with a lot of math that I am not qualified to do. So my main role was just kind of looking at the data, looking for patterns, and also writing the code that allowed us to groups.
I guess I kind of have a question with that, where it was like the picture of the car and then the attack and then the like edited picture. So how do you actually apply that middle one to the picture?
So it just kind of goes over top of the image. It basically changes the pixels that are on there. And so obviously it's very slight change so that you can see it, but it just kind of goes.
What was the last flower you identified? Do you remember?
I actually, there's still some that are left to identify from that very last trip, but I think the very last one that I have identified, second to last one was a cardinal flower, beautiful red flower, highly recommend you look it up. And then the very last one I saw was called an American Hog Peanut.
How did you all find your internship? Maybe in just a couple of sentences each.
So I was told that REUs were super important for me early on, so I applied to a couple the summer between freshman and sophomore year. Didn't get any of those, so I just again was like, okay, I'll apply to some more. I actually had another job lined up here and then got the REU, so I just found it on the NSF website.
I went into my senior study knowing I wanted to do something with plants. I took the plant biology course here. Highly recommend. I fell in love with it through that course. And I went up to my advisor, Dr. Unger, and I said, what can I do? What are my possibilities? Because my brain was very big and I needed to figure out something down to earth. Then we got on the topic of working with mountain homes. And so that's what we transitioned to doing with after multiple talks with them. And then I actually got the SQ grant over the summer that funded me to let me continue doing that work over the summer.
So I've lived in Oak Ridge, obviously, so I was familiar with the lab, and I was actually supposed to intern under my mentor, whose name was Dr. Stephen Young, in 2020, but obviously that was the year COVID hit. So then I went to apply again, but there was a gap in that if you weren't technically an undergraduate or a high school student, you couldn't apply. So finally this year, I was able to apply to the program and kind of connect with them and do work with them.
mention two things. One thing that I think is kind of cool is that as different as they seem, Jessica's project and Carmela's project are overlap in this weird way, right? Because it's that AI, you're using AI to identify these flowers, right? And Jessica's looking at how those things work and are accurate, which I think is just kind of neat and a nice happy accident about having people up there together. But also on Carmela talked about getting the grant from SQ. Those grants are still available. They will be available next summer as well. So be thinking about that if you're in the room and you want to approach a faculty member about summer work. They may have some ideas.
I'm also more than happy to talk to you guys about what I experienced, like from my own personal benefit as well. It was awesome.